And welcome back and to your next day of e-learning. In the previous class, you played a DNA structure preview and then watched a video on Crash Course from Crash Course Biology on DNA structure and replication. So today we're going to get into DNA and what it is. And then later on, we'll talk about what it does and how it's used. DNA stands for deoxyribonucleic acid. This is the most important molecule there is to life. This is the molecule that makes up your chromosomes and determines your genes. It is the way that the DNA is arranged and put together that allows it to dictate the traits that you have. So let's get into its structure right now. DNA is a spiral nucleic acid, one of our 4 organic molecules. Its building blocks are called nucleotides, and these nucleotides are held together by different types of covalent bonds. Okay, so again, the building blocks of DNA are called nucleic acids and are held together by covalent bonds. The reason it spirals up is the covalent bonds are rather weak, and the spiraling shape helps it to maintain its shape. So, how do we know of the structure of DNA? We're going to talk about 4 different people here, and the first of those people is a scientist by the name of Rosalind Franklin. What did Rosalind Franklin do? She was able to take x-rays actually of a DNA molecule, and the picture you see there is what she saw, and by looking at that and sharing her information with the people that we are going to be talking about here after her, we were able to figure out the shape of a DNA and how or what it's made up of and how it goes together. Okay, the X-shaped pattern that you see in that middle shows that it is a twisted or spiral molecule. As you can see in that, and this is how we come up with the double helix concept. Okay, the angle of the X-shaped pattern suggests that there are 2 strands. So again, it is Rosalind Franklin's X-ray diffraction allow us to discover that it is a double helix. Other clues that come from this suggest that there are nitrogenous bases are near the center of the DNA molecule, okay, which we have found to be true. And as we will talk about later then, the phosphate and sugar molecules make up the sides of our ladder, and we'll talk about that more here in a little bit. From Franklin, we're going to run into a guy by the name of Erwin Chargaff. Now, as I mentioned, there were nitrogenous bases. There are 4 nitrogenous bases that make up DNA. They are G-guanine, C-cytosine, A-adenine, and T-thymine. Chargaff came up with the first parity rule, or what we call today complementary base pairing, and what he discovered is that the percentages of guanine and cytosine are almost the same in any DNA molecule, and the percentages of adenine and thymine are almost always the same in a DNA molecule. So therefore, when the bases pair together, it is always adenine bonding to thymine and guanine bonding to cytosine. This, as I said, today is called Chargaff's rules. Okay, adenine always pairs with thymine, guanine always pairs with cytosine. We also call this complementary base pairing. A complements T, G complements C. Then we get into these 2 guys. These are the 2 most famous people. These 2 guys, James Watson Francis Crick, actually won a Nobel Prize for their work on DNA structure. Okay, and they propose the double helix model that I mentioned earlier. But if it was not for the work of Franklin, these two guys would have not been able to complete and win their Nobel Prize. They actually believed at one point, you should have learned in the video, that DNA was actually a triple helix, not a double helix. It was after looking at her X-ray, Franklin's X-ray diffraction, that they were able to see that it was a double helix model. Okay, so again, this accounted for Franklin's X-ray diffraction. It explains Chargaff's rule of base pairing and how the two DNA strands are held together. So again, we must also, even though it is Watson and Crick that received the Nobel Prize, we must remember the work of Franklin and Chargaff in discovering the shape. So again, the building blocks of DNA is called the nucleotide. There are three basic components to a nucleotide. They are a 5-carbon sugar. Okay, in this case, the 5-carbon sugar in DNA is deoxyribose, hence the name, deoxyribonucleic acid. A phosphate group, okay, which is a phosphate surrounded by 4 oxygens with a carbon chain attached. That is what the R represents. And then the third part is a nitrogenous base. And as I mentioned earlier, there are 4 types of nitrogenous bases used to make DNA. Okay, and this is where we need our nitrogen. Okay, so right here we can see what a nucleotide looks like. Right here at the top right corner is the nitrogenous base. Here in the middle is our 5-carbon sugar deoxyribose, and over here to the left is our phosphate group. Okay. Holding these things together are special types of covalent bonds, as I mentioned earlier. The phosphate group is attached to the sugar with a carbon-oxygen bond. The oxygen from the phosphate joins with one of the carbons in the sugar, and specifically it joins on the 5-carbon. On the other side, we have what is called a carbon-nitrogen bond. Okay, and then this is the nitrogen from our nitrogenous base joining with the 3rd carbon of the deoxyribose molecule. So again, we have carbon-oxygen bonds holding on the phosphates, and we have carbon-nitrogen bonds holding on to the nitrogenous base, both of them attaching to the different sides of the sugar. As I mentioned, there are 4 different types of nitrogenous bases: adenine, guanine, cytosine, and thymine. Okay, here they are pictured and their different shapes. You can see that thymine and cytosine are single rings, and that adenine and guanine are double rings, and we talked about that earlier in the year. So let's go with the structure. As I said, it is now called a double helix, thanks to Watson and Crick. We commonly think of this as a twisted ladder. Okay, it is the bases, the adenine, thymine, cytosine, guanine, that make up the steps to that ladder. And then we say that it has a phosphorus carbon backbone. Those actually make up the sides of the ladder. So again, the sides are made up of sugars and phosphate groups, while the rungs are made up of the base pairs. And if we follow Chargaff's rules, adenine always complements thymine, guanine always complements cytosine. Okay, A to T, C to G. Here you can see when we draw this how things go together. This is an actual look, if we were to think of that, of how the atoms would join together. This is a more realistic list. This is a simplified version just so you can see together. Now there are 2 different sides to this molecule, as we said, hence the double helix, and they spiral in different directions. Okay, you can see here we have 1 running from 3 to 5, and then we have 3 running to 5. Okay, so our double helix is always put together in a 3 to 5 direction. Okay, this is what we call anti-parallel strands. The 2 strands run in opposite directions, as I just mentioned. Okay, the direction here, as you can see, this direction, we're going to 5 to 3 direction this way. This one is going 5 to the direction upward. So they are going in opposite directions. They are anti-parallel. Okay, that is the only way that the bases will fit together, placing the nitrogenous bases as the rung and the phosphates and the sugars on the sides of our ladder. Okay. Holding the bases together, as you can see in this picture, are very, very weak covalent bonds called hydrogen bonds. Okay, these bonds can be easily broken, which is important when we go to using DNA and to make the special proteins we need to make that determine our traits. Okay, again, these are very, very weak bonds that once the strand is unwound are easily broken apart. Okay, this is, again, I said, very critical to DNA's function of producing the necessary proteins and determining our traits. Okay, that is the end of the slideshow. I would like you now to do your worksheet. It begins on page 344 in your text. Have a good day.